All right, let's dive in. Today, we're tackling eukaryotic gene regulation.
Oh, yeah, exciting stuff.
We're going to figure out how our cells decide which genes to turn on, which to turn off.
It's way more complicated than just a simple switch, that's for sure.
Exactly. And for this deep dive, we're looking at a college-level biology textbook chapter summary.
All about transcriptional regulation.
Heavy stuff, but seriously, this system is a big deal.
Huge.
It affects, everything.
Absolutely everything. Development, diseases, you name it, cancer.
So first things first, eukaryotic gene regulation is way more intricate than what you see in bacteria.
Oh, yeah, for sure. It's not just an on-off switch.
Or like a dimmer switch.
With a ton of settings. Think of it like a complex orchestra. Each gene is like an instrument playing its part.
And they all have to be perfectly coordinated.
At the right time and at the right volume.
Otherwise, the whole symphony falls apart.
Exactly. And this complexity is because eukaryotic cells, well, they're just more complex, right?
I mean, our DNA is tucked away inside a nucleus. It's not just floating around like in bacteria.
That's a key point. That physical separation between DNA and the rest of the cell means you need a more sophisticated control system.
It's not just simple diffusion anymore.
It's a tightly controlled process.
So instead of just flipping a switch, our cells have a whole bunch of regulation going on.
Layers upon layers.
Before transcription, during, even after.
It's kind of amazing.
The textbook mentions things like chromatin remodeling, RNA processing, and even post-translational modifications.
All right, so many levels of control.
Were you aware of all those layers when you first started studying this stuff?
To be honest, I'm still learning new things about it all the time.
Really?
It's what makes it so fascinating. There's always more to discover.
So this complexity is what makes us adaptable.
Adaptable, responsive. It's what allows eukaryotic organisms to thrive in such diverse environments.
Okay, so let's break this down. The textbook describes chromatin as the gatekeeper of gene expression. What exactly does that mean?
Good question. Think of DNA as a really, really long instruction manual.
All the genes you need to build and maintain an organism.
Right. Chromatin is how that manual is organized. Sometimes the instructions are packed away, hard to get to.
And other times?
They're all laid out, ready to be read.
So chromatin is like the filing system for our DNA and makes sure the right information is accessible at the right time.
Exactly. And the basic unit of that filing system is the nucleosome.
Which is DNA wrapped around those histone proteins.
Yep. And you often hear chromatin described as being open or closed.
Right. Was that concept hard to grasp at first?
It definitely helps to see it in action. Luckily, there's this classic study, Weintraub and Grudin, back in 1976.
They used an enzyme, DNase, to basically test how accessible DNA is in different types of cells.
Exactly. They found that the globin gene, the one that makes hemoglobinate, was only open in red blood cells.
Ah, because that's where it's needed.
And in other cells.
It was tucked away in closed chromatin. Those cells just knew they didn't need access to those instructions.
Pretty cool.
Totally. So it's not just the sequence of DNA that matters, but its physical structure too.
Absolutely. It directly affects which genes are active. And when you zoom out and look at the whole nucleus.
You start talking about chromosome territories and transcription factories.
Right. They sound futuristic, but it's a fascinating concept. It turns out the nucleus isn't just a random jumble of DNA. Chromosomes have designated areas.
Like a city, with different zones for different activities.
Perfect analogy. And these transcription factories are like hubs where the machinery is concentrated for transcribing genes.
And genes actually move around to these hubs.
They do. It's a dynamic system. It ensures that the right genes are expressed at the right place at the right time.
This is blowing my mind. So much going on beneath the surface.
And we've only just begun. There's so much more to uncover as we delve deeper into the layers of eukaryotic gene regulation.
I can't wait. Okay, so chromatin is like the filing system, keeps our DNA organized. But how does the cell know how to organize those files in the first place? Like, are there labels or signals?
You're thinking about epigenetics.
Epigenetics, right. That's the next level of control, yeah.
It is. Think of DNA as the blueprint. And epigenetics. Those are like the notes scribbled in the margin.
Annotations.
Yeah, exactly. They give extra instructions on how to read the blueprint.
So these notes, they can actually change how genes are expressed without changing the DNA sequence itself.
They do.
That's wild. It's like someone can change the design just by adding notes to the blueprint without changing the original drawing.
That's a great way to put it. These are molecular annotations. They come in the form of chemical tags.
I,
added to the DNA or to those histone proteins.
the ones the DNA wraps around.
Exactly. They don't alter the genetic code, but they can have a huge impact.
On whether a gene is turned on or off.
On or off, up or down. The textbook talks about DNA methylation and histone modification.
Right. Can you break those down a bit more?
Sure. Let's start with DNA methylation. That's a process. A methyl group gets attached to, well, it's attached to a specific base in the DNA sequence, cytosine.
Okay, and what does that do? Is it like a switch that turns genes off?
Often, yes. DNA methylation, it frequently acts as a silencing signal. It's like a do not disturb sign on that section of the DNA.
It's not just whether the DNA is accessible, but also whether there are these chemical tags, these, what were they called?
Methyl groups, in this case.
Right, methyl groups.
Yeah.
They add another level of control. Okay, what about histone modifications? What are those all about?
So those histone proteins, the ones that DNA wraps around to form nucleosomes,
Yeah.
well, they're tails,
Yeah.
the ends of these proteins.
Okay.
They can be modified by adding or removing small chemical groups.
Like what?
Acetyl groups, methyl groups.
So it's like adding accessories to those histone proteins.
I got it. And these modifications can change how tightly those histones grip the DNA.
Which would totally affect how accessible the genes are.
Exactly. It's all about accessibility. And these modifications, they can have opposite effects. Sometimes adding an acetyl group, it relaxes the chromatin structure.
Making the genes more accessible.
Right. But adding a methyl group can do the opposite. Tighten things up. Make the genes less accessible. It really depends on the specific modification and where it occurs.
This is getting complicated.
Yeah.
But it's pretty cool. So we've got DNA methylation, kind of like a silencer, and histone modifications. They're tweaking the accessibility. But the textbook that mentioned that these modifications aren't permanent, they can change. Is that right?
Oh, absolutely. And that's one of the most exciting things about epigenetics. We're finding more and more evidence that lifestyle factors, environmental exposures, all sorts of things.
Like what?
Stress, diet, they can all affect these epigenetic marks.
Hold on. So things we experience, they can actually change how our genes are expressed, even though the DNA sequence stays the same.
It seems that way.
That's amazing. It means our genes aren't necessarily our destiny.
It really challenges that idea, doesn't it? It suggests that we have some control over how our genes are expressed through our choices, our experiences. And get this,
What?
researchers are finding that these modifications can be passed down to future generations.
Yeah, we see. You mean like what our grandparents' experience could affect how our genes work today?
It's possible.
That's a big deal.
It has huge implications. It changes how we think about disease, how it develops, how we might prevent it.
And treat it.
Of course. For example, scientists are finding all sorts of methylation patterns in cancer cells.
So we could figure out how to manipulate these epigenetic modifications.
We might be able to develop new therapies.
Target the root cause of the disease. Wow. Okay, so we've got this whole epigenetic landscape. But let's move on to the next level of control.
Cis-regulatory elements and transacting factors.
Okay, so we've got the stage, that's chromatin. We've got the annotations, that's epigenetics.
Now it's time to meet the actors.
And the directors.
Exactly. So picture this. DNA is the script.
Genes are the actors, performing their roles.
But you need a stage, a director.
Lighting, a whole crew.
You got it. And that's where cis-regulatory elements and transacting factors come in.
Okay, tell me about these cis-regulatory elements. What are they exactly?
Think of them as DNA sequences. They're like stage directions. For gene expression, they don't code for proteins.
They're like instructions for the instructions.
In a way, yes. They act as binding sites for other molecules that control transcription. The process of making RNA from DNA.
So they're like signposts on the DNA, showing the cell where to start and stop.
Precisely. Some common examples are promoters, enhancers, and silencers.
Promoters are like the start here signals, right? For RNA polymerase.
Exactly. That's where transcription begins.
And enhancers, they boost the transcription rate.
Yeah.
And silencers, they do the opposite. But do they always act on genes right next to them? On the DNA strand?
You're right about their functions. But here's the thing about enhancers. They can be located really far away from the gene they regulate, thousands of base pairs away.
Really? That's weird.
And sometimes they even work in reverse orientation.
So it's like a stage director giving cues from the back of the theater.
Good analogy.
Yeah.
And silencers, those are more like critics trying to shut the whole show down.
Binding to repressor proteins that block transcription.
Exactly. And both enhancers and silencers can be very specific. They might only work in certain cell types or under specific conditions.
It's like having a whole team of stage managers making sure everything runs smoothly.
A very complex production. And the directors in all this, those are the transacting factors. They're the proteins. They bind to those cis-regulatory elements we were talking about.
So they decide which genes get to be the stars of the show.
You could say that. Some are activators. They boost transcription when they bind to enhancers.
And others are repressors. They do the opposite.
Bind to silencers, reduce transcription.
There must be tons of these transacting factors, right?
A whole cast of characters, each with its own preferences. It's a complex and dynamic system.
They respond to signals both inside and outside the cell.
They do. It's a constant conversation. Fine-tuning gene expression based on what the cell needs. To really see how this all works together, let's zoom in on a specific example. The metallothiamine 2A gene, or MT2A for short.
MT2A. Okay, what's special about that one?
It's responsible for producing a protein. That protein protects cells from heavy metals.
Heavy metals. Like what?
Zinc, cadmium, toxic stuff.
So it's a defense mechanism.
It is a critical one.
Yeah.
And it's a fantastic example of how intricate, how responsive gene regulation can be.
Okay, I'm interested. How's this gene regulated?
Well, the MT2A gene, it's got the whole works. Promoters, enhancers, silencers. The whole ensemble cast.
Okay, all work together.
Depending on what the cell needs, they can work in different combinations.
So when everything's normal, how much of this protective protein is being made?
Under normal circumstances, the expression is low, just kind of humming along. But when those cells encounter heavy metals or experience certain stresses...
That's when things get interesting.
Exactly. Transcription of the MT2A gene, it goes way up. That way the cell has enough of that protective protein.
To deal with the threat. Wow, it's amazing how all these elements work together. Chromatin structure, epigenetic modifications, cis-regulatory elements, transacting factors. It's like a complex orchestra responding to the conductor.
It's a beautiful system, isn't it? It's this interplay that allows cells to be so responsive and maintain that balance between too much and too little of a protein.
This is seriously cool. But we're just scratching the surface here, right? There's got to be more to this story.
Oh, there's always more. For example, when we start talking about how these different regulatory mechanisms interact with each other, it gets even more complex.
Interactions, like how do they actually work together at the molecular level?
Yeah.
Interactions, like how do they actually work together at the molecular level?
Well, think back to those transacting factors. The directors of gene expression, they're not just passively sitting on the DNA. These proteins are like multi-talented performers.
Performers.
Yeah, they have different functional domains. They give them their regulatory powers.
So it's like a director who can also write the script, act in the play.
And design the costumes. Haha, exactly.
What kinds of domains are we talking about here?
Well, first you have the DNA binding domain. That's what lets them attach to specific sequences in the DNA within those cis regulatory elements. But then many of them, they also have something called a transactivating domain or a transrepressing domain.
Those are the ones that actually control whether a gene is turned up or down.
Exactly. A transactivating domain, it can interact with other proteins that are involved in transcription. So it's like
Gathering the team.
exactly, boosting the expression. And then a transrepressing domain, it does the opposite. It hinders transcription, silences the gene.
So it's like, I don't know, like a director who can inspire the actors or tell them to take 5. Pretty powerful stuff.
They are. And these domains, they come in all sorts of forms. Each one interacts with other molecules in different ways, like DNA binding domains. They have these common structural motifs, things like helix, turn helix, zinc fingers, leucine zippers.
Catchy names.
They're named for their structures, their three-dimensional shapes. It's really quite elegant.
Evolution's pretty amazing, huh?
It is. But with all this complexity, well, sometimes things go wrong.
Like what?
Misregulation of gene expression. It's actually a major factor in lots of diseases, including cancer.
Cancer, yeah. Makes sense. If the balance gets thrown off.
That's when cells can lose control over their growth and division, which leads to tumor formation.
So understanding gene regulation is not just about understanding life, it's about treating disease, too.
Exactly. If we could figure out how to manipulate these mechanisms,
That would be huge.
we could develop therapies that actually target the root of the problem instead of just managing the symptoms.
Imagine that. Being able to cure diseases by tweaking gene regulation. Wow.
It's definitely a promising frontier in science. Who knows what the future holds?
This has been an incredible deep dive. We've gone from Like the basics of DNA packaging.
Chromatin.
Chromatin, right?
Oh.
All the way to the intricate interactions of transcription factors.
And cis-regulatory elements.
Don't forget those.
Hmm.
It's clear that eukaryotic gene regulation is incredibly complex.
That's so fascinating.
It really is. It's like, it makes you appreciate the sheer complexity of life.
Well, dear listener, I hope this deep dive has given you a new perspective on what's happening inside your own cells. Every single one of us is a symphony of gene regulation.
It's a masterpiece of evolution. So the next time you look in the mirror, remember that. You're not just a collection of cells. You're a walking, talking, thinking testament to the power of gene regulation.
It's true. And as we continue to explore this system,
Who knows what we'll discover?
there's still so much to learn.
Well, that's all the time we have for today.
Okay.
Till next time, keep exploring
Keep questioning.
and keep diving deep into the amazing world of science.